Please. You may have to go to the microphone because you have a very soft voice.
All right, I'm here first.
Oh.
Seize the
sorry.
moment. So, Sebastian, it was a great talk. I'm encouraged to see, how you guys are tackling, immune stimulation. What I was hoping to learn a bit more about is how are you going to be looking at platelets, thrombocytopenia, and whether there is any correlation between what you're seeing on immunostimulation and platelet effects.
Well, platelet is definitely more challenging, for sure. We had an easy time with PBMC and hepatotoxicity in the context of having easy access to models, mouse models. Is that better? Okay.
Yes, thank you.
Thank you. So with PBMC or hepatotoxicity, it's easy. You have easy access to in vitro model, a mouse model. When it comes to platelet, and especially you have to deal with primates. So the investigation of those mechanisms is much more challenging, and especially in the case of sporadic platelet decrease, trying to generate enough data to get a good grasp of what's going on is more challenging. But some of my colleagues here, Scott and Padma in particular, have invested a lot of effort, and I think making some nice progress in terms of characterizing that toxicity.
I have two questions. I think we have toxicologists on the panel. I think there's one comment is that you think that rat is more sensitive as compared to mouse and non-human primate. Is that is that sort of the consensus among the toxicologists that rat is the more sensitive species? And the second question is that, for example, recently for the neurology indication, if the drug has to be given intrathecally, we have seen that recently, for example, the Ionis, the collaboration with Roche or Ionis collaboration with Biogen, for the study are conducted elsewhere other than in the United States. So is there anything FDA either concerned or any suggestions, or we could actually have the first in human study in the US instead of in Europe for the intrathecally delivered drug to treat neurological diseases?
So for the rat, one of the issue in particular is some of the kinetoxicity and the limiting aging rat nephropathy. So it limits your ability to do more comprehensive testing. So I think from that point of view, mouse is just happens to be a better model, because of the specificity of that toxicity limited to the rat. As for your second question, I'm not completely sure to answer that one.
The second question is more
if I could just add a comment, because I am the one that called attention to the importance of testing in rat. I was specifically referring to lipid nanoparticle programs where I've seen many examples where they show much greater toxicity in rat than in mouse. So therapeutic indices estimated in mouse to be, well over 100, at least a factor of 10 difference going into rat, depends on the formulation. But a related factor is that the rat has a lot more blood, so there's a lot more bad news that you can learn if you do the test in rat.
I think my second question is also for the two speakers from FDA.
Certainly. So I think as to why the sponsors choose to do in Europe or Canada, you'll have to ask them that. We try to be flexible and understanding. Many of these rare disease populations, they're global clinical trials because you just have handfuls of these ultra orphan diseases, and being able to have enough patients to conduct a study. We also look from a regulatory standpoint, post hoc, at different centers. Is the study results being driven by, one center in Russia or something like that? And we'll have our regulatory investigators go out and look. So we do account for that, but we also understand that, these are global development programs of ultra-orphan diseases in many cases, and, we encourage the sponsors to collect as many individuals as possible.
And it's probably worth pointing out that nusinersen was approved in the US well before it was approved in Europe. So I don't see there being issues with respect to either where the studies were done or the receptivity of that particular division, despite the fact I've been burned by that division, but.
I'm referring to the first in human study, especially, for example, recently we've seen the, there are examples that for the intrathecally delivered oligos, they start in Europe instead of in the US, the first in human study. That's in patients.
Yeah, like Dr. Rogers, I can't speak to the selection of sites.
That's up to the companies. Again, I think from what I've seen with our interaction, we've been quite encouraging of companies to begin those studies here. So I don't think there's any barriers. There might be some other reasons, economic reasons perhaps, but I can't speak to that.
Over here.
All right, so my question is for Sebastian. In your model of predicting hepatotoxicity of GAPMERs, what sort of features are you using to try to predict those things?
I can't talk about that.
Thanks.
Hi. So I have a question for the panel, but also for the room in general. For targeting the liver, GalNAc seems to be kind of the consensus now. Outside of the liver, I think Dr. Dowdy was not the first one to mention this, but he's definitely been one of the most vocal. Receptor-mediated endocytosis on non-liver cells, you've got about 10,000 to 100,000 receptors per cell. So it's really difficult for, say, double-stranded RNA to get out of the endosome at an appreciable rate. If that's the case, there seems to be a lot of chemical approaches to getting out of the endosome, chloroquine or chloroquine-like, or melatonin for peptide-mediated. For a cell biologist, what's more intriguing is that these endosomes are very dynamic organelles. They mature, and there are internal proteins that get shuttled between, say, the ER and the endosomes, the Golgi and the ER. And AB5 toxins, like Shiga toxin, go from endocytosis all the way to the ER. So I'm curious why there hasn't been more talks about, say, going through a retrograde transport system to a more amenable organelle than, say, the lysosome. And perhaps being in the ER might be, in dividing cells, a more preferential place to deliver.
So I'll take a crack at that one. I've a little bit at stake there. So the point you raised with respect to endosomal escape, clearly we know that there's sufficient endosomal escape from a GalNAc transported oligonucleotide to get sufficient concentrations of oligonucleotide in the cell. To Paul's point, yes, it is less than 0.1% of the oligo that's available, but that's enough. And yes, we can see good pharmacologic activity. You can find other, and you'll hear about this from one of my colleagues at my company tomorrow, you can find cell surface receptors that will internalize oligonucleotides, and then probably through some of those pathways that you're talking about, there's enough endogenous leakiness without using a melatonin, without using another toxin, which is potentially more difficult to control what lysosomal enzymes escape from that compartment and still take advantage of the, I'll call it endogenous leakiness of the endosome. Are there other approaches that can be taken where you get uptake and go through other transport pathways? Probably, but there's nothing unique about GalNAc, and there's nothing unique about either the sRNA mechanism. We can do the same thing with an ASO. So there's the possibility of having that kind of leakage. Should we study it more? Sure, but as Paul pointed out, if you have 99.9% of the drug in an endosome, it's really hard to figure out what's the free part. So it's a chemical issue about what's free and what's not.
Just a quick response to that. With ASGR, you've got 500,000 to 1 million receptors per cell, and the turnover is about 15 to 20 minutes. So the sheer volume that you can get in there is much higher than, say, a clathrin-coated pit that's going to get in and recycled maybe every 90 minutes with a receptor at a much smaller count. So yeah, 0.01% of millions and millions is much greater than 0.01% of, say, like 10,000.
So first I need to announce that there's a young investigators meeting that starts now, and they are supposed to excuse themselves. All the rest of us can stay. And then I'll tell you that there are other receptors where the receptor number is in the range of 10,000 or slightly more per cell. That will still produce adequate knockdown.
Thank you.
Wait for Andy Gale's talk tomorrow. Thanks.
So, over. Yeah, so this is more
So
of
yeah,
a...
just a timeout. So if you're going to the young investigators lunch and discussion, this is the time.
Great. So this is more of a request for comment than a specific question, but it kind of dovetails on the last question. Paul introduced this notion of consolidation as being really healthy for a new therapeutic area in the context of antibodies. And the suggestion that we're seeing that, I think, with GalNAc conjugation for siRNAs. And I just invite the panel to comment on the flip side of that, which may be the opportunity costs that we lose in investing in new technologies for delivery, which is still such a huge thing. And how do we manage that? How do we actually incentivize investments into new technologies? How do you see the ecosystem playing towards that versus just consolidating around a clinically proven technology and deepening that? So I think anybody can comment on that, but I'd just like to get your perspectives on that.
Well, I'm certainly happy to comment on that. I think it's a really important point. The GalNAc conjugates have yet to reach the market. I think that we can anticipate that they will in the very near future. That'll be a super important milestone for our field. But a related point that I think Matt was touching on in his question is that, at least in my view, that technology is addressing a very limited fraction of the opportunity space in our field. And if all the energies go towards bringing those products to market with sort of a general feeling of buzziness that, wow, delivery is solved, we will have totally failed because the delivery challenges extrahepatically are going to be, I think, a lot more difficult. So the key question is how can the field deliver new technologies to deliver the products, extrahepatically? And I don't have a good answer for that. I think that in general, I think that it's bad news for our field that so much of the activity now occurs in small companies as opposed to large companies, which, at least in the golden days, the salad days, were in a position to invest for the long term. It's just not the landscape that we inhabit today. But as a consequence, I think that companies just don't have the time horizon necessary for these multi-decade initiatives that will be required. And I don't see anybody else. The academic groups, I think, are simply not in a position to deliver usable technologies that will address that.
And if I can just expand on that a little bit, our ecosystem classically has been academic, innovation, then small companies forming out of that, and then the small companies getting acquired by bigger companies. But in this case, the innovation really came from the group at Merck and the group at Alnylam, both working on GalNAc, not through that traditional approach, really. And so I think it's hard to tell where the new innovations are going to come from. I will say from the VC perspective, talking a lot to different VCs in the early days of RNAi, all the VCs knew that delivery was critical, and they started investing in new delivery companies. But they quickly realized that there were so many delivery ideas coming forward, but they were typically, a new lipid and a different nanoparticle. They were not fundamentally transformational, and they were innovations that, 30% increase in activity, whereas what we need are tenfold increases in activity. So I don't know where the new innovations are going to come from. There is a lot of money out there in the VC community sloshing around, and if someone does come up with something really extraordinary and it's real, I think we'll be able to fund it and develop it, but I don't know.
I just want to add something to that. I think the delivery has not been a unique issue to oligos. I think it's been an issue that spanned across small molecules, And coming from oncology, we see it even with cytotoxic agents. Now we're getting bispecifics, we're getting antibody-drug conjugates, and you know, the nice thing about oligos is that they're not extremely cytotoxic. So, you know, if you're going to conjugate them to something and try to put forward some targeted delivery, at least you have that going for you. So I think what I'd like to leave you all with is just to keep moving forward. And I heard a number thrown out there, 100 years, but in 100 years, we're going to have all new people on the earth. So please try to make your mark before that, and we'll try to review all that data, and if it's safe and efficacious, you know, approve it.
Manu?
I just want to add one more comment regarding the delivery. Of course, GalNAc is very unique in terms of the high capacity and the low affinity in terms of high affinity. But on the other hand, I think the lot of small molecules from the large pharma have not been looked at. Namely, over the years, these compounds have been developed, and for some reason of safety or something like that, they have boxed, and they have not been addressed back. And what is their really capacity, and as carriers, whether they have any function. That has not been looked at, and that can be addressed. And I think because that has been done as a function of tissue and organ, and that has a lot of hope. And that's why I think the chemists have a role to play in evaluating and understanding those molecules.
Maybe if I can take another second and give the perspective from Ioni's point of view. We certainly realize that there is opportunities beyond GalNAc, and we're deeply invested in trying to find those new modalities that will help us target muscle, kidneys, and various organs. And really hopefully in the next few years you will start seeing some of those new modalities being published and presented. But I think it's very encouraging that the progress we've made with GalNAc can be translated to other organs, leveraging new route of delivery.
Next question.
My question is about idiosyncratic toxicity events. I'm wondering if in larger studies you've seen or expect to see differences between oligotherapies and small molecules in terms of the frequency of idiosyncratic events.
Well, sort of by definition, that's difficult to assess. I think with oligos, being that many of these are for, again, rare ultra-orphan diseases, we don't have the large safety databases that they're being accumulated in the post-marketing setting, so that's yet to be determined. As far as the comparison with small molecules, I haven't any clue. Your guess is as good as mine.
All right.
I have a question to the colleagues from the FDA, and actually inspired by one of your titles, Emily, which was saying challenges to generalization. I love that. Now, in the area of antisense oligonucleotides, and Manu showed it, we are working with these stereoisomeric mixtures so far, right? And now we are able to synthesize these oligonucleotides in a stereo-defined fashion. So I just wonder what your thoughts are. Are we moving into this direction to have a single molecule in the end, or will we be making mixtures in 100 years from now?
I can't say whether or not we've had any applications in that area, but certainly if they're working, we will look at them and review them. I think that's a growing field and has a lot of potential. Did you have a more specific question? I couldn't understand exactly what you said.
I just think that when currently we are we are talking about all these issues that we are facing with oligonucleotides, and I think some of them could be solved if we didn't have to talk about mixtures of oligos. We're talking about mixtures of up to 500,000 molecules. And if it was a single molecule, we could address these questions far more specifically. So my question is, would the FDA rather suggest to move into this direction, or because it's very complicated, of course? You know, are we happy with still using this generalized?
Yeah, no, it is very complicated. I don't think we're recommending to move in any direction unless the drug is working and it's safe. So if you can provide that evidence, then move in that direction.
The fair answer would be the jury is still out there and it is not clearly established. Having said that, I want you to listen to talk from Wave and other people in the meeting, but RP versus SP, they have their own unique qualities, some attributes in terms of contributing positively. So that's where it stands right now, in my opinion.
Great answer.
Eric van der Veer from the Netherlands. For the FDA, I was interested in hearing the steps that you've made in terms of course, the large number of filings and compounds that are starting to come out, and the assessment thereof, of course, of the data that they present. I was wondering if you have any communique with the EMA in Europe. In that respect, you had mentioned, of course, there was the discrepancy between the acceptance in the US versus nusinersen in Europe, which took a little longer. And I'm wondering if there's any discussion in terms of trying to advance those two in a similar fashion as opposed to these delays.
So we have quarterly TCONs with the EMA where we'll discuss important approvals and we'll discuss our thinking and their thinking.
Yeah.
As far as the timelines, when they do things and we do things, we're
Sure.
independent organizations from that standpoint. But yes, we do discuss and we try to harmonize comments and issues ahead of time. But again, there's
Right.
At the end of the day, we're still two different organizations. But I found our discussions to be quite productive with them and sort of getting their thinking and the transparency of our thinking as well.
Okay,
And I think it's mutually beneficial.
So you're also confident of the fact that the EMA is also moving, at least trying to proceed this as quickly as possible as well, these technologies.
I certainly believe so from my
Okay,
interaction.
great.
They just have such a different process,
Yeah.
right?
It's-
It's
Yeah.
such a different process getting all the countries to buy into
Sure.
recommendations from a rapporteur.
Of course,
So it by nature can take longer.
I agree. But just interesting. Thank you.
So my second question is about ADME. And I was wondering if anyone here has seen sequence-specific differences in ADME. In particular, I'm interested in fully modified oligos, but I'm curious about any other ones.
The answer is yes. We do. Sorry, I'll take it. There are differences. Even small differences in protein binding that can be related from sequence to sequence, or even just putting a single phosphodiester into a molecule will markedly change the ADME. So again, in the early days we portrayed these molecules as having very similar pharmacokinetic profiles from one to the next. But the more we learned about it, the more we learned that there were different flavors. And the more you modified the oligo, the more we found that there were slight variations from molecule to molecule. So there are definitely sequence and chemistry differences in the ADME, even with a narrow class like single-stranded phosphorothioate oligonucleotides. But within the broad spectrum, you can generally describe a pharmacokinetic profile rapidly cleared from plasma, binding to the same old tissues, and then being cleared slowly from the individual organ. So I hope that answers your question. Maybe Bart can amplify on that.
My simple answer was going to be yes. Chemistry and structure matter. I can't speak to specific instances, but we have observed it, yes. That's about as much detail as I can go into.
So you've observed sequence specific, so the exact same chemistry, same backbone, but different sequence? And then you
Yes.
see, okay, thank you.
Hi, my name is Ken from Canada. I had two questions. First question is, what is the biggest challenge in using antibody to direct the delivery of siRNA?
I can answer that. That's what my company does. It's making the antibody.
What's the cost?
No, to be completely serious, it's finding the right antibodies that will bind to the right epitope on a cell surface receptor, get internalized properly, release the oligonucleotide at the appropriate time, like GalNAc does, and then produce the pharmacologic activity. So I can tell you, and you'll hear a little bit about it again tomorrow at 8:50 from one of my colleagues, that you can find cell surface receptor monoclonal antibody pairs that will allow you to deliver oligonucleotide payloads to various cell types outside the liver. But it is a fine dance finding the right antibody, epitope, cell surface pair that gives you the activity.
So the reason is not all antigen internalize efficiently. Am I right?
Internalization is necessary but not sufficient.
Right. So you want to amplify the delivery and trigger internalization. Right.
And as well as have the payload come out at the appropriate time in the maturation of the endosome. You should really save this question for tomorrow. I'm sorry, I don't mean to take up everybody's time.
Sure.
If there are no further questions, I would really like to thank the audience for a really good discussion section, and especially thank the speakers who have done a really nice job at introducing the concepts.